All right, so in our overview of specific immunity, we said that antigen is going to get to the B cells and the T cells, and that is going to activate a clone, like one of the B cells or T cell clones that are then going to make copies and actually do a bunch of jobs or have their jobs or whatever. So we ended the last video about talking about how B cells and T cells actually get activated, right? So we said that antigen just drains in and B cells can just respond, and they can make antibody and pump it out in the form of humoral immunity. But for T cells, they have to be presented antigens, so their receptor is a little bit more advanced, and they have to also recognize major histocompatibility complex in order to get activated. So we're now going to go a little bit more into detail about like the clones, like the genetic variation that goes into those receptors. So this is called clonal selection theory. And basically what it says is that the sequence on the genes for the human gene that codes for the receptor for B cells and T cells has a lot of variability. So essentially really how it works is you have a bunch of different genes, and there's different combinations of the genes that get coded at any one time. And you'll take a piece of this gene, a piece of this gene, a piece of this gene, or you take two pieces of this one and one of this. And it just the way that it's set up, you can have all sorts of different combinations of these genes that go into coding for building the protein receptor on B cells and T cells, such that you can have this huge amount of genetic variation, huge amount of variation in the shape of what those receptors would respond to, and they would also respond to very specific antigens. So as this is happening, like as your immune cell or your body is making all of these immune cells with all of these diverse receptors, it has no idea sort of what it's building. It's not specifically building something that would respond to antigen. It's just building a whole bunch of random things. And so what you first need to do is to figure out which of these T cell receptors or B cell receptors, any lymphocyte receptor, which one is going to respond to you. So this happens in the bone marrow and in the thymus for T cells, bone marrow for B cells, where we're actually going to screen all of these lymphocytes to make sure that the random receptor that we put on the surface of them isn't going to attack your own body, right? So you expose those B cells or T cells with the receptor to self cells, and any cell that binds to self will get eliminated and killed. So all of the other ones that have receptors that don't bind to self, they remain. So you can see that there's all this genetic variation. We have all these different shapes for these protein receptors that are just waiting to see if an antigen fits with them. So they're hanging out in the lymph nodes after they have been screened this is the process of maturation, right? So after they have been screened in the bone marrow or in the thymus, then they head to the lymph nodes and they're just waiting there for antigen to appear. So that antigen might drain in, and this would be a case where the antigen just drains in, so this is a B cell, and it just finds this B cell and it fits. It doesn't fit any one of these. So this clone right here we say has been selected, and that's the one that is going to start making a whole bunch of copies of itself and undergo what is called the clonal expansion. All right, so here this kind of just goes in and shows you that B cell and that T cell process. So they're both made within the bone marrow, right? So we have B cells on this side and T cells on this side. So there's a release of these immature B cells and T cells. The B cells go to special sites in the bone marrow, whereas the T cells head to the thymus. This is where they're going to start to develop those really hyper unique receptors, and they're going to mature where they're going to be screened to make sure they don't react to self. If they react to self, then we kill them off. If they don't react to self, then they migrate again, and they head to the thymus with their receptors on the surface, right? Ready to wait or not to the thymus, sorry, they head to the lymph nodes where they go to wait for antigen to be around. The T cells are on the inside of the lymph nodes, and the B cells are sort of on the outside of the lymph nodes, and this really is because of how they respond to antigen. So the B cells are on the outside, so as soon as antigen starts to drain in, they can be activated, and the T cells are on the inside because they need that antigen presentation. So the antigen presenting cells make it to the interior of the lymph nodes to present to the T cells. And even the B cells can be antigen presenters, so they can be on the outside here, and if they get exposed to antigen, they can just turn right around to the interior of the lymph node and, activate a T cell by presenting antigen to it. So let's go in and talk specifically about the B cell, the difference between the B cell and the T cell receptors. So B cell receptors are actually called immunoglobulin, and they have these, it's like this Y shape, and they have these antigen binding sites, which are these highly variable regions. I always like to think of it as like a stick figure with a person that's got two hands, and they've got two antigen binding sites that are very specific to something. And that's their variable region. And then they also have a constant region. The receptor is actually the same as antibody. It's just called antibody when it is secreted from the body. So the ends, so this is the little stick figure that I was talking about. And so here are your variable regions, like this is where you have that really high specificity, the antigen binding sites, and then down here, these are the constant fragments, so this is the same for every immunoglobulin or antibody of any, like, one person, right? It's called the constant fragment down here. If this is actually embedded in the membrane and is immunoglobulin, the membrane goes right through here, across the constant fragment, and these variable fragments are sort of hanging up in the air just waiting for antigen to come in. The T cell receptor, again, is a little bit different. It belongs to the same family, the same kind of protein as the B cell receptor, but it's a little bit smaller. It never gets secreted because, remember, the T cells actually migrate out of the lymph nodes and go to fight the infection themselves, where the B cells send the antibodies to do the job for them. And then it's also got a hook onto that major histocompatibility complex. So here's the binding site. So it does have a variable region, just like the B cell, and then it's got this constant fragment that is bound within the membrane. But then there's also a little piece, so like if the antigen presenting cell comes in right here, there's a piece that grabs onto major histocompatibility complex. So again, here, this slide just goes through all of the differences between B cells and T cells, and I have a whole other video that differentiates these things, so I'm going to just move on from there. All right, so really, what is that antigen? So antigen is any substance that's going to elicit immune response, and it is a part, a piece, a fragment of that foreign cell. A lot of times it's like some protein receptor, it could be some sort of toxin that it produces. It really is any piece of anything foreign that, in any size, that makes an immune response happen or activates a B cell or a T cell. So the B cells, again, I feel like I'm repeating myself, but I just kind of want to make sure we hit all of the pieces. B cells can recognize antigen that just flows into the lymph nodes from the site of the infection, and then as they respond, if they are activated, they start to make copies of themselves that are called plasma cells. Every single one of those plasma cells is going to pump antibody, which is essentially the receptor, so that it binds to whatever the antigen was, but now at the site of the infection. And then it also makes some copies of some cells that are the memory. Again, I have another video that will talk about what antibodies actually do to help, right? The T cells then, again, they need to be presented the antigen. They recognize that major histocompatibility complex. So macrophages, B cells, dendritic cells, these are all cells that are considered antigen-presenting cells, and they process a little bit of the antigen, and then they just bring it up to the T cells and say, here you go. And then the T cells say, okay, hey B cell, I see the antigen, and there's your major histocompatibility complex, so I know it's you, and you're not trying to trick me, and so they start to make copies of themselves, the cytotoxic T cells and helper T cells. The cytotoxic T cells leave the lymph nodes and go fight and kill foreign cells and infected host cells. And then the helper cells actually help in certain ways. And we'll talk about it again. All right, so again, this just says what I said on the last slide. So the B cells make antibodies, T cells make cytotoxic T cells and helper cells, and then also, of course, some memory so it could happen faster the next time. This shows that process of antigen presentation. So here is a cell. This is a dendritic cell, and it recognizes this microbe, and it's processed the antigen, and it's kind of holding the antigen on the surface, and it's going to come on over, and it's going to bind to this T cell right here. So this actually shows the process of both the antigen presentation and the recognizing of the major histocompatibility complex. So here's the T cell receptor in green there, and then the little blue blob is the antigen, and there's an arm of the helper cell that recognizes that major histocompatibility complex that is showing the presentation. So that's presenting the antigen. So it's really a two-step process, and then that T cell is activated. And so the helper T cells can actually then go off and communicate with some B cells. So here's a helper T cell that is communicating with a B cell that basically just sends chemical signals to it like, hey, make more copies, do your job, just sort of boost the B cell response. And then the B cells, of course, we got these plasma cells, we got the antibodies being pumped out by the plasma cells, and then, of course, some memory. So there's a lot of overlapping jobs and like cross-communication that's going on between all of these things. I'm going to skip past the slide because I actually have a whole nother video that just walks through step by step what the antibody actually does. There's 4 different things: complement fixation, neutralization, agglutination, and opsonization. So you can go ahead and watch that video. But then I kind of want to talk about like how does the immunity come from, and hopefully I can fit this in the next couple of minutes, right? So your natural immunity can be either active or passive. Active immunity means that you have gotten sick and you remember it. Passive immunity is actually directly getting immunity from your mother, so in the form of breastfeeding. And then artificial immunity can happen in both an active and a passive way too. So active is a vaccine that's going to elicit an immune response that's going to create some memory, whereas passive immunity would be injecting someone else's antibodies or antibodies from somewhere else directly into the person rather than getting the immune cells to perform the response. A lot of these antivenoms are actually made within animal hosts. So if you expose a horse to like snake venom, it will make antibody that can be then used to neutralize toxins in people. A lot of venoms are too toxic and they act too quickly that our immune system doesn't have time to present to have a response. Right? Let me see how many more slides I got. All right, so let me leave one more quick video. It'll probably be a minute or 2 long, and I will talk in general about vaccination, and then the types of antibodies and B cell and T cell videos are already posted.